All right, let's dive in. Today we're going microscopic, taking a look at histology, the study of tissues.
Definitely a fascinating area.
Yeah, and we've got some really cool academic stuff on this you sent over. Excited to dig in and explore this tiny world, the building blocks of us.
It is pretty wild when you think about all that's going on inside, huh?
Totally. We just kind of go about our day, but under the surface.
Yeah, it's like a whole hidden universe.
Like a watch. You just see the hands moving, but then you realize all these tiny gears and springs making it happen.
Oh, that's a great analogy.
Right.
And with the human body, those gears and springs are the tissues. Each one a bunch of similar cells all working together with some support, to do a specific job.
So like those tiny gears in a watch, tissues are what make up our organs and ultimately us.
You got it. And your excerpts actually break down the levels of organization beautifully.
Oh.
Starts with the cell, the basic unit of life. Then these cells team up, form tissues, those combine to make organs, then groups of organs make organ systems, and all that working together, well, that's the organism.
Okay, so it's like building a house.
Yeah.
Start with individual bricks, the cells.
Uh-huh.
Mortar holds them together. Specific patterns, you build walls, that's like the tissues.
Mm-hmm.
Different walls, you get a room or an organ, all the rooms together, boom, the house.
I like it. Really captures the idea, building from the ground up. And just like a house needs a solid foundation, tissues rely on something called the extracellular matrix.
Extracellular matrix,
Yeah.
ECM.
Exactly, ECM. Think of it like scaffolding around the cells in a tissue.
So the support system.
But it does way more than just support. It helps move nutrients around, gets rid of waste, even affects how cells talk to each other, how they behave.
Wow, so essential.
Totally. Without it, things would fall apart pretty quickly.
So cells are bricks, ECM is like, I don't know, mortar, beams, plumbing, all in one.
Yeah, something like that. And it's made of this gel-like substance, the ground substance, and then fibers, strong and elastic. Different tissues have different types and amounts of ECM. That's why they have such different properties.
Makes sense. But how do scientists actually study all this, get up close and personal with these tissues? It can't be as simple as just looking through a magnifying glass, right?
You're telling me. They have to prepare the tissues in a very particular way. It's pretty cool. Picture a histology lab, scientists carefully creating these super thin slices of tissue, so thin they become transparent enough to view under a microscope.
Histology lab, huh? Sounds intense. What happens there?
Well, first, they have to fix the tissue, like hitting pause on a biological process. They use a chemical, usually formalin, to stop it from decomposing, preserves it, kind of like taking a snapshot.
Formaldehyde. Wow, that sounds strong. Is it really necessary?
Oh yeah, it's key. Formaldehyde links proteins together, essentially freezing the structure in time. Don't worry though, it's all done safely in the lab.
Okay, so fixation is step one. What's next?
Then comes dehydration. They got to remove the water from the tissue. To do this, alcohol baths, with increasing concentrations of alcohol, pulls the water right out.
Interesting. So like drying fruit, but for tissues.
Exactly. It preps it for the next stage, which is clearing.
Clearing, like they literally make it see-through.
Yep. Light needs to pass through for the microscope to work its magic. They use a solvent, often xylene, and that does the trick, makes the tissue transparent, ready for the next step.
Wow, it's almost like a magic trick.
Yeah.
So now the tissue is see-through. What then?
Time to give that delicate, transparent tissue some support so it can be sliced super thin. That's where infiltration and embedding come in. They surround it with a supportive material, usually paraffin wax. It fills all of the tiny spaces.
Oh,
Once the wax hardens, it's like a tiny time capsule, perfectly preserved.
like encasing a fossil in amber.
Yeah.
But how do they get those super thin slices you talked about?
That's where a really cool tool steps in the microtome. It's like a miniature deli slicer, but way more precise. Get this, the slices it makes are thinner than a human hair.
That's amazing. So now they've got these ultra-thin slices, all encased in wax. Are we finally ready for the microscope?
Almost. One more crucial step. Staining. Most tissues, they're colorless naturally. To see the details under a microscope, gotta add some contrast. They use a mix of dyes called hematoxylin and eosin, usually just called H&E.
H&E, yeah, I've heard of that. But what does it actually do?
Think of it like color-coding different parts of the cell. Hematoxylin stains the nucleus, gives it this vibrant blue or purple color, and eosin stains the cytoplasm and collagen, a pinkish red.
So it's like highlighting different players on the field, right?
Yeah.
Easier to see what's what.
Exactly. And it's not just about looks. The way it stains tells us a lot about what's in the tissue.
I always get a little lost with the details there.
Yeah. It all comes down to charges, basically. Tissues with a negative charge, like the nucleic acids in the nucleus, are attracted to basic dyes like hematoxylin. So they appear basophilic, that bluish color. Tissues with a positive charge, like mitochondria, collagen, cytoplasm, they're attracted to acidic dyes like eosin, so they're acidophilic, reddish, or orange.
Okay, so the colors aren't just random. They actually tell us something about what we're looking at.
Absolutely. Now, H and E is the most common, but there are tons of other stains, each targeting specific parts of the cell, revealing different secrets.
Wow, a whole toolkit of colorful stains.
Yeah.
So cool. So once they've got these ultra-thin stain slices, now they can finally see them under the microscope.
You got it. And to make sure these slides last so they can be studied for years, they mount them on glass, special adhesives, preserve the whole thing.
It's like tiny works of art, these colorful windows into life itself. But I'm curious, is this the only way to prepare tissues?
Good question.
Uh,
It leads us to another cool technique, frozen sections.
Frozen sections, that sounds interesting. How is that different?
Well, sometimes speed is super important, like during surgery. They need to know what's going on right away. Instead of all those steps with formalin and wax, they rapidly freeze the tissue and use a special instrument called a cryostat to slice it.
Like the express lane for tissue prep. Does freezing affect what they can see?
It's actually really amazing. Frozen sections are great for preserving sensitive parts of the cell, like lipids and enzymes. Those can get damaged by the chemicals in the routine process.
So they can choose the best method depending on what they need to see.
Exactly. Each technique has its strengths and weaknesses.
Well, we've definitely covered the basics of tissue prep, but now I'm dying to know what kind of amazing things they can actually see under the microscope, when they look at these meticulously prepared tissues.
That's a great question. And it takes us into the world of microscopy and all the incredible insights it provides. But I think that's a story for part 2 of our deep dive.
Ooh, a cliffhanger. All right, everyone, stay tuned. We'll be back soon to explore those wonders waiting for us under the microscope. And we're back. Before the break, we were hanging out in Histology lab, learning how scientists get those tissues all prepped and ready for the microscope.
Yeah, it's quite a process, but totally worth it for the incredible things we can see.
I was just thinking, for centuries, we could only study the human body with the naked eye. Then the microscope comes along and boom, a whole new world opens up.
It really is amazing. And it's still happening. Histology is constantly changing, new techniques, new technology popping up all the time.
Ooh, okay, I'm all ears. What kind of advancements are changing how we study tissues?
One of the biggest is digital pathology. Imagine those tissue slides, but instead of a traditional microscope, they're scanned, turned into high-resolution digital images.
So scientists can view all those details on a computer screen instead of peering through a lens.
Exactly. It makes collaborating so much easier. Pathologists can share those images instantly with colleagues anywhere in the world, get second opinions or expert input.
Wow, like telemedicine but for tissues.
Uh-huh, yeah. And it's more than just sharing. Digital pathology allows for incredible analysis. We can use AI now to help identify patterns, anomalies in those digital slides.
Wait, AI is being used in histology labs.
Oh, yeah. It has the potential to help make diagnoses faster and more accurate.
That's incredible. What other game-changing technologies are on the horizon?
Another exciting area is three-dimensional imaging. Traditional histology gives us these flat images, but tissues are complex, three-dimensional structures.
Right, like trying to understand a mountain range from just one photo. You miss the depth, the scale.
Exactly. But new techniques like confocal and light sheet microscopy are letting us create 3D reconstructions of tissues.
Whoa, from flat images to 3D models. What kind of insights can we get from that?
Oh, so many. We can track individual cells within a tissue, see how they interact with each other in 3 dimensions, even visualize how blood vessels and nerves weave through those structures.
It's like being able to step inside the tissue and watch it all in action.
Pretty much. And this isn't just for research. 3D imaging is starting to be used in clinical practice, too, helping doctors diagnose and treat diseases more precisely.
All this talk about cells and structures makes me think of that extracellular matrix, the ECM. How do these advanced imaging techniques help us understand that better?
Great question. You're right, the ECM is just as important as the cells, crucial for tissue development, function, even disease. With these new imaging tools, we can actually visualize that intricate network of fibers and molecules.
So not just static images anymore, but dynamic interactions, like going from a still life painting to a high-def video.
I like that analogy. These insights are already leading to amazing new discoveries. For example, researchers are developing drugs that target specific parts of the ECM to treat things like cancer and fibrosis.
It's mind-blowing. The future of histology seems incredibly bright. But with all this powerful technology, are there any ethical considerations?
That's a really important question. As with any powerful tool, there's always the potential for misuse. It's crucial to have open discussions about these ethical implications as the technologies are developed.
We need to make sure these technologies are used responsibly for the benefit of everyone.
Exactly. Fostering a culture of responsible innovation in histology, prioritizing transparency, accountability, inclusivity.
Sounds like we're on the verge of a whole new chapter in how we explore tissues, one that requires a lot of thought and careful consideration.
Absolutely. The microscopic world is full of wonder and complexity, and as we dig deeper into its secrets, we have to do so responsibly and ethically.
Well said. This has been an incredible journey from the history of histology all the way to the cutting edge of scientific innovation. But before we wrap things up, I have one last question.
Shoot.
We've talked about so much, the routine process, frozen sections, digital pathology, 3D imaging, all these advancements. But are there still limitations to what we can see and understand at the microscopic level? Are there still mysteries waiting to be uncovered?
Absolutely. While we've made huge strides, there's so much we still don't know. For example, we're only just beginning to understand how genes, proteins, and the environment all interact to shape how tissues develop and function.
So even with all our fancy tools and techniques, there's still that sense of wonder and mystery in histology.
For sure. That's what makes it such an exciting and dynamic field. Every discovery leads to more questions, more challenges. It's a never-ending quest for knowledge, and one that has immense potential to improve human health and well-being.
Inspiring to think about all the discoveries that are yet to be made.
Isn't it? And who knows, maybe someday you'll be the one making those breakthroughs.
Well, after this deep dive, I'm seriously considering taking a histology class.
Uh-huh. I wouldn't blame you. It's truly fascinating.
It is. And to our listener, we hope this deep dive has inspired you to look at the world, even the world within you, with a fresh perspective. Even the tiniest structures hold incredible beauty and complexity.
Absolutely. Keep exploring, keep questioning, and keep diving deep into the wonders of life, big and small. We'll be back after a short break to wrap up our exploration of the amazing world of histology.
And we're back for the final leg of our microscopic adventure. We've been on quite a journey through the world of tissues, learning how scientists prepare them, how they visualize them, and even how technology is pushing the boundaries of what we can see and understand.
It's amazing how much we can learn from these tiny structures, isn't it?
It really is. But before we wrap up our exploration, I want to circle back to those different types of staining techniques you mentioned. We talked about hematoxylin and eosin, but you said there's a whole spectrum of other stains that can reveal different secrets hidden within the cells.
Absolutely. H&E is like the basic toolkit, but there's so much more. Your excerpts actually highlighted a few other interesting ones.
Oh, cool. I'm always fascinated by those colorful images.
Well, for instance, there's the Fulgen reaction. It's specifically for staining DNA, so scientists can see the genetic material in the nucleus.
Wow, so it's like seeing the blueprint of life itself.
Exactly. And then there's the periodic acid shift stain, or PAS. That one highlights carbohydrates, which are important for energy and cell signaling.
So it's like putting a spotlight on the sugar molecules.
Yeah.
Each stain adds another layer of understanding, like peeling back the layers of an onion.
I love that analogy. It's so true. And your source material also talked about the importance of frozen sections.
Oh, right. We touched on that earlier. It's the fast track for tissue prep, right?
Yeah, when speed is key, like in surgery, they skip the long routine process and freeze the tissue, slice it with a special tool called a cryostat.
So surgeons can get information about the tissues right away. It's like having a live feed from the operating room.
Exactly. Plus, frozen sections are good at preserving certain parts of the cell that can be damaged by the chemicals used in the routine process. Things like lipids and enzymes.
So it's all about choosing the best tool for the job.
Exactly. And with every new technology, our view of the microscopic world just keeps getting clearer.
This whole deep dive has been an incredible experience. I feel like I've gained a whole new appreciation for the amazing complexity of our bodies, all these tiny parts working together. It's mind-blowing.
I'm so glad to hear that. It's truly fascinating, isn't it?
It is. And to our listener, we hope you've been inspired to look at the world around you and inside you with a newfound sense of wonder. Even the smallest things can be incredibly beautiful and complex.
Keep exploring, keep asking questions, keep diving deep. Until next time.